name is Aidan Daly. I have a DPhil in computer science, and I'm a senior computational scientist at the New York Genome Center, a member of Himali Fetnani's Center for Genomics of Neurodegenerative Disease, as well as Sonia Vikovic's lab. And among these two labs, I look at basically ways of modeling gene expression and cellular activity in space, particularly in large cohorts of tissue. The reason I do this is because we're interested in sort of how pathology presents itself in space. We know that whatever the pathology might be, whether it be something very, natural like senescence or something maybe a bit more sporadic like ALS, the idea is that it could affect, in theory, the tissue in sort of three essential ways. One is which in the architecture of the tissue, in terms of like what cell types are present in what spatial arrangement, and if the disruption of that has led to some, has led to the pathology that is presenting. The other might be in terms of how cellular function is affected. So not what cells are present, but what a cell is doing, and if that is aberrant from its normal state. And then these two things might even happen in concert. So the last thing might be how cells across different cellular programs may be involving many genes in a single cell type, or many genes across multiple cell types are changing in concert, maybe producing an emergent behavior, or maybe responding to a common environmental cue. So kind of how do we use data from different modalities to kind of parse what's happening in a given pathology? Yeah, so some of the things that we look at, our ideal sort of way we could get at this is if we had a perfect spatial modality that was spatially resolved, obviously, so we could see what's happening in space for this pathology, that had cellular resolution, so we can pinpoint individual cellular drivers, and that had full coverage of the transcriptome or proteome or whatever ohm is being considered. So obviously, when we are looking at a pathology like senescence, that might have a very, there are some key genes that have been implicated, but we are kind of curious that as cells senesce, how different processes are affected. The wider net we can cast, the better. So we kind of have a little bit of each of these with the different methodologies available. If we're looking at barcoded capture techniques like Visium, this is a common sort of platform for looking at spatial transcriptomics, where you can look at the total transcriptome as it varies in space on a single tissue section, but generally at a coarser resolution. The sort of modern high definition versions are getting more and more resolved, where you might even get into cellular or subcellular resolution in terms of how you visualize things, but they'll never be as good as, say, fluorescence imaging, where you can zoom into whatever sort of depth you want and get a really, really fine-grained picture of what's going on, albeit with a pretty limited panel. You can only look at sort of a couple dozen to maybe a couple hundred targets if you're really lucky, depending on the technique. And then finally, there are sort of single cell methods where you can tear everything apart and look at it in a really fine-grained way, but then you've lost the spatial context of what was all next to each other. So sort of my essential questions for this research are then taking these multiple data modalities that are all incomplete views of the tissue, and then seeing if we could find like the latent state and identify what are the key drivers of pathology. And the way that we get at these sort of key drivers is looking at really large scale analysis. So not just multiple partial views of one or two tissues, but dozens or hundreds, to the point where we can say with some sort of statistical certainty what are the changes that are seen between group A and group B, where group A might be diseased and group B might be control, or we might have maybe a time resolved where we have multiple groups at different time points, and just identifying sort of common factors of spatial change, either in terms of tissue composition, cellular function, or both. And then sort of the last sort of more practical consideration is whatever tools we develop, we would like them to scale with the increasing size of data that are available and the increasing, spatial resolution or the increased coverage of new technologies that are being developed. So the genesis of cSplotch was, we had a lot of these large ST data sets looking at various pathologies, and we wanted a principled way to be able to look under the hood of what's going on. And as a motivating example, cSplotch can be applied, I will show, I hope, to many tissues. But as a motivating example, I'll talk about the colon. We were drawn to the colon because it's composed, when you look at it under the microscope, of very visually distinct cell types. So it makes it easier to address that first question of what might be changing in terms of tissue structure. And that was sort of the genesis of the question here. We know that the colon serves, an important digestive function. And we also know that as an organism develops and eventually as it starts to age, the function establishes itself in terms of its stereotypical organization, but eventually that organization starts to break down. And as you enter this sort of aged state, normally the colon maintains a sort of continually replenished inner lining that is established by basically a population of stem cells at the base of these sort of invaginations you'll see here called crypts. And these stem cells will differentiate and will move up to replace the lining as it is depleted. And the sort of continually being able to do that is essential for normal function. As you get to sort of aged condition, this starts to break down in ways that were not really fully understood. There are lots of sort of single cell studies of what happens with aging, but we don't really understand how the sort of cells interacting with their neighbors gives this sort of emergent loss of behavior, really, that leads to the breakdown of the lining of the colon, which leads to microbial infiltration and inflammation, and eventually puts at higher risk for pathologies like colon cancer. So sort of understanding the way in which these things break down in space was really intriguing for us. So the way we did this, we were working with a data set on mice where we have a lot more genotypic control, so we can remove maybe genotypic variation as a source of unwanted variation. Just focus how genotypically identical mice age. So we grew a cohort of mice, sacrificed them at various times, and profiled the colon along a couple different axes. We got samples from the more proximal colon, which is closer to the stomach, and the more distal colon, which is closer to the anus, because we hypothesized that theoretically different regions of this organ might age in different ways. And then we took sort of sections of the colon at these three different sampling locations. And we saw that, as I said, there's a stereotypical organization that it should maintain even throughout the whole life, which is sort of this muscular layer on the outside, the sort of lining that I was talking about previously on the inside, and some transitional states in the middle. So we were able to kind of annotate all the observations we had from our barcoded ST in terms of what tissue type they were, so we could zoom in and say, okay, this region is measuring, the inner lining, this region is measuring the muscle. How are these different tissue types within this sort of transverse direction changing, both in terms of cellular composition and function? So we had 11 total time points going all the way through, from birth through the aging in the 2-year window. We had the 3 colon regions, total of 65 mice and 1,500 about tissue sections. And then, like I said, this sort of distinct tissue typing scheme that we use to share information across tissue sections, to basically look at a tissue type in section 1, and then again in section 2 and section 3, and then, looking eventually at a group scale, time point 1, time point 2, time point 3, with multiple replicates at each one. And then the goal would be, can we find out how the cellular composition and how the cellular function of the colon was changing across time in all these different tissue compartments? And we also took some single-nucleus RNA-seq data, just again to have another modality. So this is where we took some of the colon and dissociated the cells and did some sequencing. So we had an idea of what the different cell types were doing in a broad sense. Like, what are the goblet cells generally doing, and how is that generally changing with age? And then we can zoom in and look at specific spatially resolved data. So overall, cSplotch worked in 3 main steps. So the first was we zoomed in on a certain region of space, and we used visual information to say what cell types are here. Like I said, the colon is a really good a really good organ to look at for this, because even under H&E staining, which is very basic, just showing basically the location of DNA with the blue stain and of protein with the kind of pinkish or purplish stain. So a very sort of basic histological staining. We could already tell apart 5 or so main visual cell types. So we could go in. You can count them all up by expert vision. We ended up training a computer vision model to automate this task just so we could process this huge data set more effectively. Then we use this visual information to guide a further tearing apart. So like if I say within this region, oh, it's mostly this purple cell type, well, I might know that this purple cell type is really something like an interstitial cell. And an interstitial cell might be comprised of a lot of different sub-cell types that I know should all look like this, but they all might be transcriptionally quite different. So looping in our single nuclear RNA-seq data, which says like, what does every cell type look like in a really kind of general sense, we could get a more fine-grained map of what cell types might be present in a given spot. So we have an estimate of what cells are in a given area of space that's informed both by visual features and by the total transcription that we observe. So that's the first step, is in inferring what cell types are in a given spot. The second would be that we model now the gene expression, the expression of any given gene at a given point in space, as a kind of combination of these cellular signatures that is also informed by covariates of interest, such as age, colon region, or sex, and tissue type ID. So if we then learn these models, we take these sort of proportions as known and we try to learn these rates, we could then, in our third step, find out which of these gene expression terms are changing a lot along a covariate of interest. So we could zoom in, for example, on a given gene and see within a spatial niche which cell types are predicted to be expressing it more highly. So this shows a gene that would be highly expressed in goblet cells but lowly expressed in T cells. If we zoomed in then and we say, okay, within the goblet cells, how are these genes changing in space? So if a goblet cell is in this sort of apex region of the colon crypt, is it expressing this gene differently than in the base? If we move, instead of this moving through the colon crypt, we might move at an organ scale and say, instead, are goblet cells in the distal colon behaving differently in the proximal colon? Or ultimately, most interestingly, how does age affect this? Are old goblet cells in a certain niche expressing genes differently than young goblet cells? So we were ultimately characterizing covariate-driven changes in gene expression, where the covariates, in this case, the most interesting one is age. And I can show a couple results from this specifically. So we looked at how first cellular composition and also gene expression were changing along this proximal distal axis in the organ, where again, the proximal is closer to the stomach and the distal is closer to the rectum. So we can see here that certain cell types are showing significant regional variation in abundance, with say like more goblet cells more distally, which makes sense. The sort of role of producing mucus for fecal compaction is more distal focused program. So it's good to have more goblet cells doing that here. But you could also see that even the goblet cells that are in a certain region are changing. They might all express certain marker genes, like TFF3 is like a quintessential goblet cell marker that is always highly expressed. But distal and proximal goblet cells express different genes, like MUC2, which is a mucus production gene, or SPR2B, which is antimicrobial gene, differently. You can look at this at other spatial axes too. We zoomed in on the one that we were really curious about, the sort of crypt axis, where in the subcrypt is where those really important stem cell populations are, and the apex of the crypt is where the lining is. So we could see the sort of gradient of cells as they move with these kind of the stem cells and muscle cells towards the base and the subcrypt, with these sort of specialized lining cells like goblet cells and colonocytes towards the apex, which is what we expect. This is all at the adult time point. So looking at a single point in time and looking at a spatial variation in gene expression and cell abundance. Here's how gene expression changes along that gradient as well. But the most interesting thing was obviously when we went into the time dimension, where we were actually looking for those multicellular programs of genes. So taking the output of the cSplotch model and trying to find sets of genes across cell types that were showing a coordinated change with age. So looking at our up here, we saw like a set of genes, this multicellular program or MCP in the subcrypt where those stem cells, those all-important stem cells live, that was showing a precipitous decline with age, particularly in the proximal and middle colon, but not in the distal. So this said to us, there might be differential effects of aging, like we supposed, depending where in the organ you are. So more in the proximal middle towards the stomach, we found this decrease of in the expression of genes spanning multiple cell types, goblet cells, fibroblasts, macrophages, as well as transit amplifying and stem cells. So we found basically an increase in genes related to inflammation and malignant transformation, and a decrease in genes related to like DNA damage repair. So it kind of implicated maybe a signal of stem cells that were not dividing correctly, and were surrounded by a lot of inflammation. And we thought this could be maybe a really cool, like sort of cancer risk signature that would be, need a lot of extensive validation on, but this kind of shows the power of looking at these things in space, where we can say we've looked at, coordinated changes of sets of cells and the genes that they're expressing within a specific spatial region of intact tissue. At a high level, we did a real big characterization of the spatial structure of the colon. We, estimated how cell composition was changing across two spatial axes, the sort of distal proximal and this sort of like outer to inner gradient. We looked at sort of the gene specific, not only the changes in the cellular composition, but cellular function in terms of gene expression. And then ultimately we found genes that were changing within these niches over time, and sets of genes even spanning multiple cell types that were showing coordinated change together. And the way that we have to consider scaling these to, new data would be that while we have been relying on older ST technology that has a pretty coarse spatial sampling across individual tissue sections, maybe only a few hundred data points per tissue section, we are now at the point where we could get thousands or even tens or hundreds of thousands of data points per tissue with sort of Visium-like or HD Visium-like technologies. So making sure that these inference methods can scale with that increased sample size. And like I said, we also have to kind of think about how we automate the data registration pipeline. So we're not actually going through and manually circling all these cells and saying, this is that cell, this is this cell. When you're dealing with millions of cells all of a sudden, that's no longer feasible. So automating tissue annotation via deep learning, I've written some code to do this, accelerating the inference using some sort of, instead of a Bayesian inference, which we have been using, moving to an approximate Bayesian inference, just necessitated by the increased size of the data. And then finally, what's really, I think, also important is making sure that all these workflows are really robust and deployable by multiple groups. So rather than just having something that works on our compute cluster and nowhere else, we have a sort of cloud compute enabled version that anyone could clone and try out themselves on their own data, even if they don't have a super high performance compute cluster available to them. So this could hopefully like democratize the methods and make sure other people can apply them to new systems of study as well. So yeah, while I'm illustrating here a particular application to the colon, the same sort of procedure could be applied, and we have indeed applied it to brain tissue, where all you really need is, for your multi-tissue ST data set, you will need a set of tissue types that are conserved across this data set that any sort of region could be reliably assigned to. This is the basis for sort of integrating this data set and sharing information across tissue sections. So if you're looking in the cortex, these could be the cortical layers, or if you're looking in the spinal cord, this could be, white matter, the horns, dorsal and ventral, anything that you can use to reliably register these data. Then within that, you would want a way of visually typing cells. So within an H&E image, this is actually a little more difficult in central nervous system because it's not as apparent what the different glial subtypes present as under H&E. But with the advent of like Visium IF, or if you have some sort of multiplexed IF imaging in general, that would be ideal. You could actually skip the guesswork of what's there and have an actual IF marker panel that says like I have designed maybe a dozen probes in this sort of multiplexed IF experiment that's going to highlight cell types I really care about and really want to make sure I call correctly. And if you have that, that's fantastic. And then you can, pretty accurately count what cell types are in a given region. And then your estimates that you'll get from the cSplotch model, which remain unchanged from this point, will just be of, greater accuracy because you won't have as much label error as trying to guess what a cell is from an H&E image. So I would say this method would only really get stronger the more we are able to kind of get highly informative visual information paired with this spatial transcriptomic data. And I think it's an open question of whether that will be resolved by new technologies that come forth or maybe a approach that can use, maybe sequential sections where you have one tissue processed with multiplexed proteomics and one processed with transcriptomics or any other omics really as it were, where the proteomic panel is designed to tell what cell types are there, and then the other subsequent section has all the information that you want to try and infer these covariate driven changes